So I'm starting. Today I will talk about Hardy-Weinberg equilibrium. So has anybody of you heard about Hardy-Weinberg or population equilibrium? Have anybody of you heard about it? Yes, no? Okay. Hardy-Weinberg?
So,
Okay, okay. So this is actually what you learn in Hardy-Weinberg equilibrium. What you learn in Hardy-Weinberg equilibrium is essentially population. It's a part of population genetics. So you deal with population over here. So when you deal with population, it's a group of individuals living in a particular space, and not everybody in a population they are look the same. Just hold on. Hey mama, একটু ভেতরে যাও, আমি একটু পড়িয়ে নিই। Okay, so when you talk about population, you can say you talk about individuals, and no two individuals look the same because there are minor genetic differences among themselves. So how do the genetic difference come? Why do some people have black eyes and why do some people have brown eyes? That is because the allele for eye color is different. Alleles are contrasting form of the same gene. The gene is for eye color, and the contrasting form are say brown eye color or blue eye color. These are the contrasting forms. In a population, you have different green eyes, brown eyes, black eyes, blue eyes. So all of them are contrasting form of the same gene, which is for eye color. So like this, basically you have a, in the population, you have a collection of these alleles. And you can see that the brown eyes are maximum. So you can clearly say that the frequency of brown-eyed people is much higher because brown eye is higher. So, but the frequency of blue eye is less. Frequency of green eye is even less. Frequency of gray eyes even more less. So this is a particular aspect in a whole population. So what you study in this population equilibrium, you study a hypothetical population where everybody, each individual, each male is capable of mating with a female, and this mating is random. It is not on the basis of religion, caste, age, whatever, relationship, whatever. It is not based on that. Everybody has a chance. It's a theoretical, hypothetical, theoretical population. So you have a random population. This is a Mendelian population where all the individuals in this population have gotten equivalent chance. All the males have gotten equal chance of mating with the females. Similarly, all the females have gotten equal chance of mating with any male. There is no bias that I like this person, therefore I'm going to mate with this person. It is not like that. It is random. So this is the assumption of you start taking. So this is known as a Mendelian population where random mating takes place. And in this particular population, there is no natural selection which takes place, no genetic drift, no mutation, no migration. All that they don't take place, provided this particular population follows this particular criteria, this Mendelian population, it follows a pattern which is predicted by the Hardy-Weinberg equilibrium. In reality, such random populations don't exist actually, but if you see the frequency of a hypothetical population, if you see the frequency of a hypothetical population, still it is not, the frequencies are not very far from the real population. For example, blood group. You know that humans have got three blood groups. One is blood group A, one is blood group B, and one is blood group O. So you have three blood groups. So, and this is responsible, it is responsible, it's a multiple allele. There are three genes, there are three alleles. One is for which gives blood group A, one which gives blood group B, and one which gives blood group O. So there are three alleles present. And you can see if you have a theoretical population, if you calculate by using Hardy-Weinberg formula, if you calculate, it is not very the percentage of people having blood group A and the percentage of people having blood group B, percentage of people having blood group AB, and percentage of people having blood group O. This percentage can be converted into frequency. You can also talk about it as frequency also. It is more or less similar to the theoretical population. So let us start this topic. I'll start by sharing the screen. I can tell you this is a very important chapter because I am expecting that, or rather hoping that one question comes from this. Normally, it used to come in former times during the offline days. This was a compulsory question in the evolutionary biology paper. So as you can see, this is a species. This is a species. Is my screen visible?
Yes, sir.
Yes,
Okay.
sir.
Okay. Inside this species, there are several populations. This is one population. This is another. This is a population. This is the second population, third population, fourth population, fifth population. So this is a population of a particular species in this world. And in this species, you have several individuals in a population. This is one individual. So this shows the relationship among species, population, and individual. Clear?
Yes, sir.
Yes, sir.
Okay. Three people are very much architects who are the architects of this modern synthetic theory of evolution. Among them, the one was Sewell Wright. The second one was Ronald Fisher. Third one was JBS Halden. They are the famous population geneticists. Population genetics. Basically, you study the transmission of genes in a population. In genetics, you see from parents to offspring. It is not, it is individual case. But here you are thinking about the same thing in terms of a whole population. Among them, Hardy and Weinberg, two individuals. Independently, they came to a law known as the Hardy-Weinberg law. We will talk about this law very soon. So, one more thing which we must need to understand, that is gene pool. What is a gene pool? Let us look at figure 14.4. So, what is the definition of gene pool? First, it is the sum total of all different allele in the breeding members of a Mendelian population at a given time. That is the gene pool. So, in this is a Mendelian population. This is a Population. Okay, this is a population, and this is an individual. Now, in this individual, A3, A4, A1, A4, A2, 1, 2, there you can see four types of alleles: A1, A2, A3, A4. You can think of this as eye color also. A1 is brown eyes, A2 is for blue eyes, A3 is for green eyes, A4 is for gray eyes. Four colors. So sum total of the frequency. So frequency means if out of 10 individual, two individuals have got blue eyes, so 2 divided by 10, 0.2 is the frequency of blue individuals. This is what you mean by frequency. So this is the frequency. So all the sum total of all the frequency of all the alleles together in the of that particular gene, A1, A2, A3, and A4, that is the gene pool. Clear what is a gene pool?
Yes, sir.
Now we come to the next definition. Here, I've talked about this right over here. We are coming to the what is genotype frequency. It is the percentage of different genotypes found with the population. So let us hypothetically think about a population, a particular gene in a population which has got two alleles. One is capital A and the other is small a. Both of them are the only two alleles present. So in a population, you have individuals who are homozygous for the capital A, capital A. They can be heterozygous, capital A, small a, one from father, one from mother. They can be small a, small a. Are you clear about this?
Yes,
Yes,
sir.
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 বুঝতে পারোনি? Yes.
Yes, sir.
Okay. So these are the three different types of genotype. Genotype means it has got one coming from father, one coming from mother, and both of them they form the genotype. Capital A is the genotype, and if you think the frequency of capital A in the population. So if you think of a population of 1,000 individuals, out of that you have 450 are capital A, 400 are capital A, small a, and 150 are small a. Then what is the genotypic frequency? Genotypic frequency of capital A is divided by 1000, that is 0.45. Similarly, for heterozygous, 400 by 1000, that is 0.4. And frequency of this is 150 by 1000, that is 0.15. And if you take the total is 1. Clear?
Yes, sir.
Okay. Now, the next thing is allelic frequency. What is this allelic frequency? It is the proportion of different alleles of a gene in a population. So, what is the frequency of the allele only capital A in the whole population? This is what you have to calculate. This is allelic frequency. Genotypic frequency is frequency of individuals who are homozygous for AA or heterozygous for A, small a. This is the genotypic frequency. But when you talk about frequency of this or frequency of small a, this is what you talk about allelic frequency. So how is allelic frequency calculated? It can be calculated by two methods. First is simply by counting genes. Are you able to see the screen?
Yes, sir.
Yes,
Is
sir.
your mobile horizontal? I think you should be able to see. If you're watching from the laptop, it should not be a problem. First is simply by counting the genes, and the second is from genotypic frequency. So in the first method, the number of alleles of one type at a particular locus is counted, and then it is divided by the total number of alleles at that locus in the population. This method is called gene counting. Remember that in a locus means capital A. So that same point is the locus. So allelic frequency equal to number of an allele at a given locus in the whole population. Remember, this is you're talking about this in the whole population. You're talking about this in the whole population, and you divide it by the total number of all alleles in that locus. Now the homozygotes are capital A, capital A or small a, small a. They are the homozygotes. Have two of the given alleles. These homozygous people, AA, they have two capital A, and heterozygotes have only one. And since total number of alleles is twice the number of individuals in a population, because one person has got two alleles, that can be AA 1 and 2, or it can be AA small a 1 and 2, or it can be AA 1 and 2. In any case, one individual has got two alleles. 10 individuals will have 20 alleles. 100 individuals will have 200 alleles. Similarly. So, allelic frequency can therefore also be counted as 2 into count of homozygotes, number of total alleles. Homozygote has got two of those alleles. Heterozygote has got one allele. So that 1 into count of heterozygotes. You add them. Then you get the total number of alleles of at a given particular locus, and you divide it by 2 into total number of individuals. Always remember this formula. Here, are you clear so far?
Yes, sir.
Okay.
Yes.
Okay. Now let us look at these two alleles, capital A and small a, and the three genotypes A. Same thing. 450, 400 heterozygous, and 150 AA, and total is 1000 individuals. You go by 1000 individuals. So frequency of A is 2 into count of AA, yeah, by heterozygous AA, right? The same formula, you're applying the formula. So you're doing 2 into 450 plus 400, 1 into 400 divided by 1000 people. So 2 into 1000. So total is you get a ratio of 0.65. You can calculate this or the frequency. This is the frequency of the capital A. 450 into 2 plus 400 divided by 1000. You apply the formula, you get 0.65. If you do 1 minus 0.65, you get 0.35, because sum total of capital A and small a together will be 1. Frequency of A plus frequency of small a, that is always equal to 1. You can calculate it by this way, or you can 2 into count of small a, capital A small a. So 2 into 150 plus 400 divided by this formula, you will also come to the same part. So this is how you calculate the allelic frequency. Second method is based on genotypic frequency. Suppose you are not given this data 450, 400, 150. This data is not given. Then you use this frequency of a given allele is frequency of homozygotes for this allele plus half of the frequency of heterozygotes of this allele. So what you have been given actually is this data. 0.45, 0.40, 0.15. You don't have the total population. You have been given this particular data. Frequency of AA equal to 0.45. Frequency of capital A small a is equal to 0.40, and frequency of small a is equal to 0.15. You have been given this data. Now from this, if you use this particular formula, frequency of A is frequency of AA plus half into this, that is 0.5 plus half into this, you get the same thing. Frequency of A, it is 0.65. You can get this by 1 minus frequency of A, you will get frequency of small a. You can use this, or alternatively, you can calculate it by using this formula 0.15 plus half into zero point 0.4, that is 0.35. So in the exam, you will be able to calculate genotypic frequency and allelic frequency. Yes, no?
Yes, sir.
Practice these sums. I've sent you this thing from this book only. Practice this sum. This is taken from Life. Author is Sanjeev Chattopadhyay. This is one of my favorite books. You can read about evolution, animal behavior from this book. You can buy this book. This is a very good book. So there is a problem. Let us calculate genotype and allelic frequencies with a real trait. In a human population, there is a chemical known as phenyl thiocarbamide. We have got a particular alleles. Some people are tasters. They can taste this. They have the genes for tasting. They can taste this chemical. And some people cannot taste. If I don't find any taste in this chemical, this is controlled by our genes. They are a group known as non-tasters. Non-tasters. They can feel the taste. The tasters can feel the taste. Non-tasters cannot feel the taste. And they have a single gene difference. The two alleles. Capital T, the capital T is for the tasters, and non-tasters is small t. So as usual, there are three genotypes. There are three genotypes in human population. Capital T, capital T small t, and small t small t. Now, capital T, like Mendel, is dominant over small t. Capital T is dominant over small t. So, in a population of 500 individuals, 225 are capital T, 150 are capital T small t, and 125 are small t. Now, your task is to find the genotypic frequency or genotype frequency and then the allelic frequency. So just simply 225 by 500, 0.45, 0.3, 0.25, 1. So you can easily calculate for this 225 divided of frequency of capital T divided by 500, that is equal to 0.45. Similarly, you can calculate for the heterozygous and the homozygous also, so the recessive homozygous. Now you can calculate the allelic frequency by putting the formula 2 into count of TT plus count of 3, 2 into 225 plus 150 divided by 2 into 500, 0.6, and 1 minus 0.6 is always 0.4. Or you can calculate it like this formula. You can use the other formula also. Since you have found out 0.45 and 0.25, you can calculate this again, you get the same answer. Clear? Yes? No?
Yes, sir.
Okay.
Yes,
When
sir.
this is clear, now we will move on to the principle of Hardy-Weinberg equilibrium. Right now, we have learned the basics of how to calculate allelic frequency and genotypic frequency. We understood what is allelic frequency and what is genotype frequency. Now, we will understand the laws. What will be the what will be the what will be frequency of AA, frequency of A small a, frequency of AA in a population. What will be the frequency of A and frequency of small a? What type of law will determine? This particular frequency? What type of law will be present? So these are other sums which I've given. You don't have to study all these things. Now we come to Hardy-Weinberg. G.H. Hardy was a British mathematician at Cambridge. I have been to Cambridge, I know, near London. And Wilhelm Weinberg, a German physician, Stuttgart. I have been to Stuttgart also. Both of these cities I have been there. So Hardy and Weinberg. I have seen the Hardy's laboratory also in Cambridge, and I have seen Weinberg's office also, or you can say his chamber in Stuttgart. They are all kept as monuments there. They independently published paper, and together their laws are known as, they describe the mathematical relationship between allelic frequency and genotypic frequency, a mathematical relationship. And this is known as the Hardy-Weinberg principle. So it has got two parts. First is a set of assumption and the second is a set of prediction. Main statement is in an infinitely large randomly mating population which is free from mutation, migration, and natural selection. First thing, note carefully, the allelic frequency do not change from one generation to the other. That is, if random mating takes place, each male has got equal chance of mating with each female. There is no preference, random. যে যাকে পাচ্ছে মেট করে নিচ্ছে, something like this. If in this particular population, where there is no mutation, nobody moves from this population, migrates from this population to another population, or natural selection or survival of the fittest, one allele finally dominates and becomes dominant, that doesn't take place. The allelic frequency which you have seen for testers and non-testers, zero point 0.6 and 0.4, from one generation of this population to the next generation, it will remain the same. It will not change. Right? If out of 10 individuals, 6 individuals are testers, in that population, it has to be infinitely large. That is infinite, as large as possible. The larger it is, the more accurate it is. But in smaller population, this doesn't work. You have the Sewell right effect or the right effect, genetic drift taking place. But in an infinite large population, this doesn't take place. So zero point 0.6 and 0.4 or zero point 0.6, the 6 individuals will always, in my father's generation also, 0.6 individuals will also, 0.6 individuals out of 10 will have, will be testers. In my generation also, 6 individuals out of 10 will be testers. In my children's generations also, it will be the same. The allelic frequencies will not change. The second prediction is the genotypic frequencies in the population can be determined in a predictable way by the allelic frequency. So, let's open a whiteboard. This is, I'll just write down the assumptions clearly. Hardy-Weinberg equilibrium. So the first part is the assumption. Population must be infinitely large. Right, then random mating. As you can see, this is actually very theoretical. Actually, in practically, it will not happen. Theoretical. But you need somewhere to start your mathematics. There should be random mating. No migration. No mutation. And no natural selection. Because once natural selection starts to take place, your allelic frequencies will change. So when if you take this assumption, the next thing will be your prediction. What are the prediction? Allelic frequency will remain same from one generation to another. Second is the genotype frequency will depend upon allelic frequency. Now, let us consider two alleles in a population, capital A and small a. You have males, male population, you have a female population. Males have gametes, A. So let the frequency of A be P and frequency of small a be Q. So FA is equal to P F small a is equal to Q. So let's do a Punnett square. From Punnett square, you can understand. In the males, you have two alleles, capital A in the population, gene pool. Capital A, small a. Then you have females below, capital A, small a. So males will mate with females. When they meet, when these two they meet, it becomes capital A, capital A. What will be the frequency? P square. A into A. Probability of A is P. Probability of this A is P, or the frequency. So this will be P square. Similarly, A into small a over here, it will be PQ. Again, A into this, it will be PM into PN, your the frequency, or just multiplication of frequencies. This will also be PQ. And small a, it will be Q square. So what would be the total, the actual frequency of AA? It's simple, P square plus 1PQ, 2PQ. 2PQ plus Q square. And this is nothing but P plus Q whole square. The funny part is frequency of A is equal to P and frequency of small a is equal to Q. One thing is P plus Q is equal to 1. So P plus Q whole square will also be equal to 1. So the sum of the genotype frequency in a Hardy-Weinberg population is 1. Clear so far?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So this is what I've actually talked right now. Now, let us look at this table. Results of a random mating in a population having the genotypic frequency this, and this, which I've given. These are the males on the top. AA is equal to P square, 2PQ. AA is equal to Q square. Now, these are the females. Right? So now, in this population, these males are mating with these females. Now, two genotypes. This is a random mating between genotypes. At that time, you saw mating with the alleles. You're considering what the genotype is. So, from here, one thing I've forgotten to mention. From this, you can see that the genotype frequency P square for frequency of AA, 2PQ for frequency of AA, and Q square for frequency of AA. So these are the genotypic frequencies. These are the genotype. This dependent on the allelic frequency. Right? This dependent on the allelic frequency, which was frequency of A is equal to P, frequency of small a is equal to Q. So in a Hardy-Weinberg population, the genotype frequency depends on the allelic frequency. Also, one more thing which I mentioned, one more point. A population which is not in Hardy-Weinberg equilibrium, if you give one generation of random mating, you will immediately, the population will come in Hardy-Weinberg equilibrium. Now this we will explain right now. So now this is a, like I said, a random mating. A random mating আকাশ হয়ে গেছে. Random mating taking place between the genotypes AA, AA, small a. This is males and females. AA, these are the females. So if this is P, AA is P square, we have seen 2PQ plus Q square. If you multiply this, this becomes P to the power 4, this becomes 2PQ Q, P square Q square, 2PQ this. Like this, if you multiply them, this will become, this value comes. So result of this meeting, so capital A into capital A meetings, this is capital A meetings. So like this, all these meetings are being done. They have been putting them in a table. So you get this particular formula. All the types of meetings which have taken place, you are putting them over here. You get this total Q square. So this actually, if you solve this problem, this also becomes P plus Q whole square whole square. Whole squares whole square. And that is also equal to 1. 1 square square, that is also equal to 1. So you can say the allelic frequency determines genotype frequency and the genotype frequency remains the same in all generations. I hope you are clear about this. Yes, no?
Yes, sir.
Okay, yes,
Yes, sir.
Yes, sir.
So you people have not fallen asleep. So this is the same thing they have done with the testers and non-testers. They have solved this problem and they have found that the genotype and allelic frequency will remain in the same in the next generation. It will remain 1 in all generations. So the next is question which can come is, one generation of random mating restores Hardy-Weinberg equilibrium, which I just taught right now. So now this is a founder population. This is the frequency is not in equilibrium. AA is 0.45, capital is 0.25, and this small a is 0.35. This is not in equilibrium. If you find the allelic frequency using the formula P, frequency of A is equal to P, and frequency of small a is equal to Q. If you use the formula of calculating allelic frequency, you will find that A is 0.55, capital A, and small a is 0.45. You can calculate. This is it. In the next generation, if random mating takes place, the sperms, eggs, the AA is equal to P square. So P square here P is equal to 0.55. This is 0.3025. Capital, this is 0.2475. This is also 0.2475. And Q square is 0.2025. So you can find this formula. So the genotypic frequencies equal to 1. And now if you calculate the allelic frequencies, you will see it remains 0.55 and 0.45. They remain unchanged. So one generation of random mating, it will restore the Hardy-Weinberg equilibrium. So I hope it is clear today. বুঝতে পেরেছো? Any questions?
No, sir.
Okay, very good. Then next class, maybe we will take it on Tuesday, online class. On Thursday, we'll have an online plus offline class. Okay?
Okay, sir.
Yes, sir.
Okay.